Hello, I'm Scott Sorrell here at the ADSA meetings with Kristin Gallagher from Michigan State University. We got a Spartan with us today. Welcome, Kristin, to the Real Science Exchange.
Thank you so much. Excited to be here at ADSA in Kansas City.
Yeah, awesome. And my co-host today, Dr. Clay Zimmerman, the trusty Rusty Clay Zimmerman, and Carrie Estes. Kristin presented a poster yesterday. The title of her poster was Abomasal infusion of branched-chain ketoacids alter lactation performance in early lactation cows. Kristin, tell us a little bit about the study.
Yeah, of course. So this was a USDA NIFA project that we worked through this past winter. And we were looking at abomasal infusion of branched-chain amino acids or branched-chain ketoacids in the early lactation period and its effects on performance and liver health, specifically looking at its relationship to fatty liver disease. So this project, the major results from this project were that branched-chain amino acids altered milk yield, increased milk yield, milk protein yield, milk fat, but we did not see any changes in liver triglycerides as opposed to the branched-chain keto acid infusion altering and efficiently decreasing liver triglycerides.
Oh. It's interesting that you infused it abomasally instead of jugularly.
Jugularly, yes. So yes, trying to do long-term jugular infusions turned out to be a bit of a challenge, but our main objective was to deliver these amino acids and keto acids post-ruminally, and we're going to be analyzing plasma branched-chain amino acid concentration and other amino acid concentration to see how that is going to be affected. But yeah, so we delivered these to the animals post-ruminally, and we got some pretty significant results that we're excited about.
So I noticed that you infused very specific amounts of each of the amino acids. So can you tell us how you came up with those levels?
Yeah, absolutely. So we infused the branched amino acids are valine, leucine, and isoleucine. And with this study, our objective was to increase plasma branched-chain amino acids by approximately 30%. And that comes from some work done by Dr. Zhou in some previous research where he established there was a relationship between animals in the early lactation period that he classified as having high liver triglyceride versus low liver triglyceride. And in those animals with low liver triglycerides and really a lower risk of fatty liver disease, he found that there was an elevation of plasma branched-chain amino acids by about 30%. So that's where that came from. And to determine if the concentration of amino acids was correct, we did a preliminary trial where we had 6 cows of the same cohort that we were able to effectively increase branched-chain amino acids by that amount, so by about 30%.
So Kristin, I think probably a lot of our listeners are familiar with feeding or infusing branched-chain amino acids. Mm-hmm. What's the hypothesis behind the branched-chain keto acids?
Right. So again, we're interested in the liver's response to these amino acids. And unlike other essential amino acids that we might be delivering to the cow, branched-chain amino acids are not going to be metabolized in the liver. Most of the amino acids, over 80% of the amino acids, are instead going to be metabolized by peripheral tissues, so muscle and adipose tissue, and it's going to be partially catabolized to these keto acids, which are a non-nitrogenous, non-amine metabolite of these amino acids. And there, these branched-chain keto acids will then be taken up by the liver, and they can be used as an energy source. And we have some cellular data that might show that these branched-chain keto acids will affect hepatocyte metabolism. So that was our target and our interest, was to understand the relationship between these branched-chain amino acids and keto acids and how they affect liver health and production.
So you saw quite a response in production to the branched-chain amino acids.
Yes, yeah. So we definitely saw a pretty big milk response that we're pretty excited about. And I will comment again that the diets or these treatments that we deliver to the cow, we fed the same diet, but different treatments, were not isonitrogenous or isoenergetic. So in our branched-chain amino acid treatment, again, these are amino acids with amine groups. We were feeding higher RUP to these branched-chain amino acid supplemented cows. So I think we had, just more nitrogen available for milk production in these treatments, as well as there's some literature that goes to suggest that some of these amino acids we're feeding, such as leucine or isoleucine, can increase anabolic activity within the mammary gland, potentially increasing mammary plasma flow, activating the mammalian target of rapamycin, mTOR. So we think that there is, just delivering more RUP, but also doing some more anabolic work as well. And with the keto acids, we didn't see any difference in our milk production response, but again, we saw an effective decrease in liver triglycerides. So that was exciting.
So with your liver tag, you saw the reduction with the keto acids,
Yes.
right? But
Yep.
not necessarily with the branch chain.
Yeah, not with amino acids. So, so we're going to kind of continue to look at the mechanisms that are going to be occurring in the liver. We have tissue that we're going to be looking at gene expression relating to lipid uptake and how these keto acids might be affecting lipid uptake, beta oxidation. My lab mate, Izabelle Bernstein, is going to hopefully be coming out with, we'll all be coming out with a lot of future analyses from this project. I'm excited to really go through and better understand what's happening with our plasma amino acids. We just sent those samples out last week and are hoping to get a lot of information back from that. Looking at antioxidant status of the liver, as well as we have tissue samples from muscle, adipose, and more liver tissue that we're excited to analyze.
So, Kristin, I know soy protein is high in branched chain amino acid content. So, is it possible to formulate a diet today that might deliver more branched chain amino acids?
Yeah, of course. So branched chain amino acids are really important in dairy nutrition in general. They make up over 20% of the total amino acid profile in milk. There's a lot of these branched chain amino acids that are going to be found in different forages and different protein products in our TMRs. For example, we just fed a common Midwest diet that was 50% forage to our early lactation animals, and leucine was about 1.3% of the total dry matter versus some amino acids we might be familiar with. Methionine was about 0.29% of dry matter. So even without supplementing it, just on our base TMR that was being delivered, I think what we need to be wary of is how these branched chain amino acids are going to be fermented in the rumen. Because they will become branched chain VFAs and they can have, different end products once they're fermented in the rumen. So certainly if you if you can balance for what is going to be delivered post-ruminally in these high protein products, you could potentially establish similar results if you really wanted to increase branched chain amino acids, plasma branched chain amino acids. It would certainly be a challenge, but could certainly be done. Meanwhile, the branched chain keto acids, this is again a metabolite, so we don't currently have a way to feed those branched chain keto acids in its pure form. So we'll see if any technology is going to be applied to that in the future.
So I'm kind of curious, Clay. We're talking about practical implications. Would we need to, maybe this is a better question for Kristin, but maybe think about protecting them ruminally with...
Yeah, so I mean, there is still, I think, a lot of work to be done to figure out, when we should be supplementing these branched chain amino acids and what they might be able to do for different herds and different dairy farmers. But I think that it certainly is there's a good opportunity that these could be things that we're balancing for in the future, and that delivering these post-ruminally can be very helpful to the animals and to milk production and health responses. So I think that's definitely a very promising subject to better identify.
Yeah. So you're going to be finishing up your thesis. You're going to be moving on. Are you going to be handing the ball off to anybody else upon your departure?
Yes, absolutely. So yes, I'm a grad student in Dr. Zhang Zhou's lab at Michigan State University, and I had a co-lead on this big project. Her name is Izabelle Bernstein, and we have a lot more work to be done with this project, and are super excited to better understand the mechanisms behind branched chain amino acid, branched chain keto acid metabolism. So I'm hoping to get into the industry after this, and we'll have a lot more information coming from our group.
Very well. Maybe we'll have Izabelle in this chair next year.
I hope so, yeah.
So what are your plans after you graduate?
So after graduation, I'm excited to just get back in the industry and work in technical sales and consulting, and really just help farmers and make better decisions for the herds out there.
Okay, and I understand you may want to go back to New York.
Yeah, so I'm originally from central New York, not too far from Syracuse, and headed back to the East Coast, northeast, so we'll see what happens there.
All right, very well. Anybody looking for a very bright, very energetic master student? We've got one here in Kristin, and so give her a call. Kristin, thank you very much for joining us today.
Absolutely. Thank you so much for having me.
Very well. We're here at the American Dairy Science Association meetings where we've been interviewing students and talking about the research that they've been conducting over the last year or so. With us today is Dr. Tom Overton. Tom, you're not a student anymore.
I am not a student.
But Allison Kerwin was the young lady that did the research. Now we're going to be reviewing 3 separate abstracts that Allison worked on. First, tell us why Allison's not here.
So Allison had her second baby back in the wintertime, and so
Nice.
She's kind of finishing up her maternity leave. She's been taking that kind of in phases and decided that maybe Kansas City and 100-degree temperatures with a 6-month-old
great
wasn't really the
decision.
great plan, so it probably makes sense.
Yeah, congratulations, Allison. Wish you could be here. Tom, we've got 3 abstracts that we've kind of lumped together. Going to talk about those. Can you kind of give us an overview of what they were?
Yeah, so Allison, for her PhD, conducted, one of the, I would think, probably the largest scope transition cow management studies in commercial farms that's really been done. 72 herds in New York and Vermont. Visited larger freestall dairies. She visited each dairy 4 times, and as she likes to remind me, drove about 58,000 miles in the course of doing the data collection for this study. So it's a large study, a lot of data, and we're starting to roll it out not only within Journal of Dairy Science, but also obviously here at the meetings.
Yeah, excellent. Now, Tom, I usually have one co-host that joins me for these podcasts. Today, for somebody with your level of expertise, I had to have 2. So with me, I've got Dr. Marco Zanobi and Dr. Jeff Elliott, and I talked about a lot of data to go through. I'm going to leave it to these guys to kind of dig into the data with you. So, gentlemen.
So actually the first question I want to ask goes back a few years, maybe to grad school days. I want to know why you knocked my dog's tooth out.
Yeah, I really feel badly about that. We were roughhousing a little bit, and I do feel bad for Dutch.
Yeah,
But he's quite a.
I still
quite a
remember the
dog.
tooth just flying away.
Yeah, I remember other things in graduate school too. If you'd like to talk about those, or
No, we need to skip those stories.
Okay.
What was the idea behind this big project? Why you wanted to do this? I mean, what is the story behind?
Yeah, so going back into the kind of the 2005 to 2008 time frame, we did a large study with Daryl Neidham. Daryl Neidham was the lead at Cornell. Our group was collaborative on it and also very involved, in which we looked at 100 herds, looked at relationships of NEFAs and blood ketones with outcomes on dairies. And so we had some nice data from that data set that really, or results anyway, that kind of provided the basis for how we do cow and herd level NEFA and blood ketone testing these days. But one of the things we realized at the time is we really didn't do a very good job or a very thorough job at characterizing all the nutritional and non-nutritional factors that can contribute to herd level success. And so, we really wanted to take this opportunity to do a much more in-depth characterization of those factors. And so, Allison, along with our collaborators, Buzz Burhans and Daryl Neidham, again in this case, as primary collaborators, we were really focused on what we wanted to try to collect, both from the standpoint of nutritional strategies of herds, but also the various things that we know at the pen level and at the herd level, contribute on the non-nutritional side, to outcomes. And I think we're much more sensitized to those relationships now than we were then. And so that was kind of how the study was born. Was an attempt to do a much more thorough job and then try to relate some of those management practices or other characteristics at the herd level, to our outcomes and also to productivity and reproductive function and herd health.
Okay. So, can you share with us the main result, the main important result from this?
Yeah. So I think there's a few different things to share. 2 of the papers are now published in Journal of Dairy Science. So we've published now the data set, right? So people can see, really what the characteristics of the herds were in all these different areas. We published the second paper; those are all now available, where we looked at again, kind of covering some of the same area that we did before relative to relationships of things like blood NEFAs and ketones with our outcomes of interest, and results were quite similar to the previous work, and so that part, we feel good about that the recommendations we've been making for the industry kind of still hold. The one unique part of it was we actually looked at inflammatory markers, especially haptoglobin as a marker of inflammation, in those cows. in the data set, and probably not of surprise to people. We associated, and again, these are all associative things. These are epidemiological studies. We associated higher levels of haptoglobin postpartum as a marker of systemic inflammation with more disease, with poor milk production or with poor reproductive performance. So that kind of extends that data. Again, adds to our growing knowledge of inflammation in that postpartum cow and what it might mean at the herd level.
So Tom, on that, the result was high forage resulted in higher levels of haptoglobin, I believe.
Well, I think it depended, right? I mean, there was some of that. Again, with the epi data sets, they're always a little bit of, you always got to think through the interpretation. I mean, some of the things I think, especially with the nutritional side, are very consistent with the literature as we know it, right? So cows fed a more, controlled energy diet, a lower energy diet, during the prepartum period, had lower blood ketones, postpartum, and that fits our knowledge from the control literature. The haptoglobin story, I think, is still a bit interesting in terms of not really fully understanding how diet can affect that. And so both prepartum diet and also postpartum diet. I think there's parts of it that are clearer than others, but there's still more to.
And that's where my curiosity comes in. Actually, on Friday, Marcos and I had a webinar with a customer, and not related to this abstract at all, but he says, haptoglobin, the correct, or is it the most accurate biomarker that we need to be using, or is it a cytokine or another messenger?
Yeah. So I think that area is still kind of wide open, honestly. We chose haptoglobin in part because we had previous data that we'd done at the commercial farm level where we were able to, in a smaller data set, but still, 400 cows worth of data associate elevated haptoglobin with negative relationships with outcomes. That was some of Julie Huzzy's PhD work in our program. There's other markers. There's serum amyloid A, another acute phase protein. Both those tend to elevate in especially with ruminal or gut type inflammatory processes. But again, I think the thing with acute phase proteins is they'll elevate, with any kind of systemic inflammation. Cytokines, I think those are all, those are all on the table relative to how we might, think about relationships with outcomes and things like that. And again, it's associations. I think one of the things that gets a little bit, if you lock a bunch of immunologists or others in a room for 24 hours and tell them to come up with one marker to measure immune function, right? They'll probably debate it for 24 hours and probably still won't come out with any. I mean, again, and I always say, I don't say it to make any, to make fun of anything other than just to convey it's a complex area, right? And complex to interpret. And so I think, we may end up with a suite of markers at the end of the day that we may look at some of these things for something that gives us a little different look than we get out of our energy-related analytes like NEFAs and ketones and things like that.
So to change gears a little bit, to go to the more nutritional strategies.
Yep.
What did the findings here tell us about feeding starch relative in the close-up period as well as the fresh cow period? Because y'all have done a lot of work there over the years. Yep. It's always been intriguing.
Our data set in this case, actually, that one was one of the clearer things in that, cows fed the higher starch diet post-calving anyway, did have lower blood ketones, and so, that would appear to be an advantage. One of the things that was, was also interesting is that, we did not pick up any clear relationships with milk yield, right, for any of the kind of nutritional strategies we looked at. I don't think that means that it doesn't matter, right? I think that's probably a bit of an oversimplification.
I was going to ask that, because that's the way it comes across. But I
realize that, right? But I mean, at the same time, I think there's, I think one of the things to remember here, especially when you're looking at epidemiological data set, is, we're making choices in terms of how we look at the data, okay, and across a range of outcomes. Now, if we do a controlled research study, right, I can compare this level starch with this level starch and be far apart, okay, and be very different diets. Well, that's not the real world, right? Here you're looking at, and I'm not going to say a continuum because we're trying to target lower or higher in certain cases, but commercial farm diets don't vary as much as what you're going to come up with in a controlled research situation, right? If Marcos here were to do a controlled study, he's going to really look to define some treatment differences and do that, right?
Yep.
So, so I think that's part of it, right? So again, I certainly don't think it doesn't matter. We clearly have enough, information from the controlled research, right? And of course, you guys have meta-analyses on, on choline. There's meta-analyses on DK, not put all back on meta-analyses, right? But that clearly show, production relationships with some of these nutritional strategies, right?
Yep.
And so again, we do these epi studies in part to, extend the control research knowledge, right, or try to see how it plays out in the real world, but then also generate hypotheses that we might go back and test.
So I will go back to the close-up pen. A simple question. So there was no association, although, of NDF level with any of the outcomes, but there was an association with meal yield and probably break rate with pen state particle separator. So how we can interpret that data? Sorting and probably.
Yeah. So again, I think that's one of the areas where that's obviously of interest now. Far off, right? So I know you're talking close up, right? But far off, there was a positive association with, more in the top screen, leading to better outcomes in general. I think that, to me, I wonder if that actually represents more forage level, feeding level in the diet, right? One of the things you have to be careful of in these studies is things move together, right? So you have more forage in that diet, we're gonna have more probably in the top screen. We feed less grain in those diets, right, typically. So, and again, if you do the math, the magnitude of relationships actually is not huge, right? So you gotta keep that in mind. Close up, again, you're still looking at relatively small differences there at the end of the day, and so I think you got to keep that in mind. I think for us, we still like if you're looking at particle size of chopped straw or hay, that 1/3, 1/3, 1/3 in each of the three sections, or you add the bottom two together for a third in the 4-section Penn State box. 10 to 20% in the top screen, less than 40% of the pan in the bottom two. For close-up diets, I think you can be a little coarser with far-off diets and be okay, just because you don't have the same level of grain in the diet.
Right.
So.
Okay.
And of course, I think we all know feeding management of these diets, is also really important, right, from a particle size and moisture standpoint.
So moving to the fresh pen.
Yep.
There is a nice, I think, that association with NDF, undigestible NDF, and effective UNDF with PREC. So that's pretty important, I think. Do you have any sweet spot where you want to be in the fresh pen?
So one of the things, you're kind of quoting some of the digestibility. So the PEU, the undigested NDF content of the diets, the physically effective, and so basically as those increased, we saw lower milk yields in lactation. To me, that just mostly comes back to just forage quality, right? So if you think about poor quality forages, right, they're going to have more UNDF, they have more PEUNDF, and so if we're feeding poor quality poor quality forages from a digestibility standpoint, we're going to have likely less milk yield there. In terms of recommendations, I think one of the things that's tricky on that, and why we haven't really published a lot of recommendations on the UNDF or PEUNDF levels in diets, and we've done some of this work separately, has been the models aren't always fully seeded in terms of that data, especially for the non-forage fiber sources that might be in diets. And so what I tend to tell people is, if your typical UNDF 240 level in a lactating diet, high cow diet, for example, is around 7. You might run that fresh cow diet if you have one up around 8, maybe a little more. But I think any more than that's probably going to hold those cows back a bit, because we have had definite experience in our research group trying to do work in this area where we think some additional fiber for these fresh cows is beneficial, but it's very clear that you could hold them back if you go too far with that, especially if you go for longer. I think that about 10 to 14 days with a higher fiber strategy, especially higher forage fiber strategy in fresh cows. So that's something I think we're still kind of wrestling around with, but I think it's got implications for how we feed fresh cows going forward.
Tom, we're shoving a lot of data into a short period of time. If you were to summarize it for the audience, what would be just 1 or 2 key takeaways from the research so far? And then how do you see going forward with the research?
Yeah, so thanks, Scott. So I think, again, from a big, broad standpoint, I think both nutritional and non-nutritional factors come into play when we're looking at both transition cow outcomes, where there's disease or disorders, to some extent repro, and obviously milk production. Although again, we didn't see, at least nutritionally, in the big broad brush stroke, didn't see a lot of relationships there. I think our data support the efforts of the industry to decrease stocking density, especially the feed muck in these fresh groups and also pre-fresh groups. I think in general, our data also support that heifers, will do better if they're not commingled in this time frame. And I think also there's certain things on for example, group changes before calving. Herds that move cows less. In other words, no more than like a move to a close up and then a move to calve. That appeared to be a better strategy in the data set than herds that move cows more frequently than that. Added an additional move or two. And I think that I think some of those things are consistent with what we think we see when we work with herds out in the industry, right? We see that herds that streamline those things, herds that pay attention to stocking density, herds that herds that those cows don't linger too long in the calving pen. I think that plays with what we see.
Mm-hmm.
Okay,
So Tom, a lot of those things, maybe the guys around this table take as common sense now. We've talked about it. More water, don't let them linger, plenty of space. But those farms that aren't doing that, is there a reason? How do we get them to make those simple, I guess I'm calling them simple adjustments. I don't know if they are, maybe they're not on those farms.
Well, I think again, I think they just need to keep hearing it, right? I think from as many sources as possible, right? And we've been doing that, right? I mean, it feels like we've been doing that. I think that comes back into play. More information that continues to support, or the recommendations that a lot of us make, I think is helpful. And again, I think it's just part of that constant coaching that always happens in the dairy industry, whether it comes from, when you guys are working with farms, for example, right, with what you do, to nutritionists, veterinarians, others out in the industry, extension making, directly interfacing with herds.
Tom, thanks for stopping by here at the Real Science Exchange. It's always great to see you.
No, pleasure.
Look forward to seeing you next time.
Okay, sounds good. Thanks, Scott.
And we're back here at the American Dairy Science Association meetings. This session has a couple themes. One is Brazil. We have two guests from Brazil, and the predominant theme is Virginia Tech. So we've got three people here that spent some time at Virginia Tech. So that's kind of cool. We're going to be reviewing a paper by Yumi Taguti. Did I say that right?
Yeah.
Close enough? All right,
Yeah,
very well.
good.
And she's an undergraduate. She's visiting from Brazil. And she presented a paper. Was it a paper or a poster?
It's a poster today, yeah.
Yep. Called Linking Amino Acids to Milk Fat Synthesis. So, welcome. Can you tell us a little bit about how you got the idea for this research?
Yeah, sure. So I think the first thing we need to understand is how milk fat is important, because milk fat is what drives the milk price in the industry. So knowing that the milk fat yield in cows supplemented with amino acid has changed, we had like in vitro studies, previous in vitro studies about how the mTOR pathway acts behind this milk fat yield changes, and this was realized that this was conducted in mammary epithelial cells in supraphysiological levels. So what our goal now is to understand how is the relationship between the essential amino acids and the milk fat synthesis and physiological levels.
Oh, very interesting.
Yeah.
And I see you brought a guest with you today. Is this your professor?
Yes.
Yeah, Izabelle Teixeira.
Perfect.
Close. All right.
Excellent.
Now, yeah, that confused me just a little bit because you're no longer at Virginia Tech. You're currently at the University of Idaho, but you were there doing a sabbatical.
Yeah, it was in 2017. I was a faculty in Brazil at Unesp, and then I went to do a sabbatical at Virginia Tech.
Okay.
When we were discussing, Mark Hennigan and I, about the project, the possibilities, we came out with this idea just to evaluate the role of the essential amino acids on the milk fat synthesis. And the very first experiment study in this sense in our group, I mean, my group and Mark Hennigan's group,
Yeah.
was in that time.
Yeah.
So it was nice, and we could figure out some amino acids that were playing a role because we were omitting amino acids on the media on the epithelial cells, mammary gland. So, and we could see that when we were omitting some amino acids, it really shut the de novo milk fat synthesis.
Ah.
So I stayed 2 years. It was a 2-year sabbatical. I went back to Brazil, and currently I'm at University of Idaho. I just joined University of Idaho.
Okay.
So that's a long story, and are joining me through those all changes.
Yeah.
So,
Well, we're happy to have you here with us today
Thank you.
at the Real Science Exchange. My co-host, Carrie Estes, once again, you've been here your third time. So I'll hand it off to you. I think you got some questions.
Okay, yes. So I'm not an expert at all in cell culture. I'm just going to set that up right now. But I was curious about the stage of lactation that you took the mammary tissue from. What stage were they at?
Okay, so I think the most important thing that we considered when we were taking the tissue samples was to get from a cow that is a high producer animal, but I think mostly in the mid lactation.
How did you define high producing? I'm sorry.
High producing for in the farm that we're working currently, high producing will be around 50 kilograms of milk per day.
That's high.
During 2 milkings. But I was not involved in the cell isolation process because I was not here yet. I think maybe Izabelle can tell you more about this part of the process.
yeah, so we did some because we had some papers back in that time, and I think you'll join some. I sort of overlap with Carrie at Virginia
Yeah.
Tech. So anyway, every time you were reading papers about isolating cells, it was not very clear. So we were able to establish at Hennigan's lab a way for isolating cells. So I started doing that with biopsy samples. That was nice. That's why we were able to get like middle lactation because we want to be sure that the cells were able to secrete milk components, because that was important for us and also for other studies that Mark were running at the lab at that time. After that, after I went back to Brazil, Alex Burberry also refined a little bit more the technique. But just being direct to your question, so mid-lactation is good, and we want to be sure that that cells are secreting milk components.
Yeah, it becomes pretty important then.
It is.
Yeah, sure.
Yeah.
Sure.
Yeah.
And with those cells, we were able to check that they were secreting protein and fat.
Okay.
So we could address different questions with the same cells.
So then once you have your cells and you're applying the treatment, how long do you let that treatment work on those cells before you end up taking your sample?
So what we did, we started with 23 hours of starvation on the cells, so they would not get overloads. And after that, we applied the treatments for 4 hours. So we were using labeled acetate as a tracer, C13. So the period that we need to let the treatment stays in the cells is more about it's related to the period that the acetate enrichment increases and it reaches the stability. So Izabelle conducted a study before to know when was the increase. When was the best time the enrichment of C13? So after this research, we could choose 4 hours for the treatments. Yeah.
Okay.
Yeah, it depends on the kind of questions, right, Carrie? Because, for example, if you are doing Western with those cells, we cannot go, if you want to do to check the phosphorylation, generally we go up to 1 hour and a half maximum because we want to see the phosphorylation that is a very fast process. With the isotope, because we want to see the enrichment of the cells with C13, we want to get is still up the curve, not reaching the plateau. So we chose 4 hours. Generally, that's what we're getting the best. But Yumi also used with a second trial that she's running right now at Virginia Tech. We just figured out that perhaps it would be better to have the cells exposed a little bit more. So she exposed the cells to 6 hours, right?
Yeah, 6 hours this time.
Yeah. So as we are, because sometimes just can get confused, is it enough time? Because we are focusing on mechanism and really understand the mechanism. So we need to address those questions accordingly. That's why we are using a short period of time.
Yeah, makes sense. Makes sense. So you did a lot of this work, very interesting work in vitro. What about in vivo? Do you guys have plans to move this into the cow?
Yeah, we do. So the first, we had 2 in vitro experiments until now. In the first one, we were analyzing the individual effects of each essential amino acid. In the second one, we could see it's still an ongoing experiment, but we can see the different levels and interactions between the amino acids that show most effect in the first experiment. And after that, we are moving on now for the third experiment that I'm doing here. It's an animal trial. So in this case, we are infusing the animals with the amino acids and trying to get an effect on the milk fat yield. So we still don't have any results about that, but yeah.
It's in the works.
Yeah, it's working.
Okay.
Let's see.
Okay, very good.
So you don't have any results on that specific, but some of the previous results, can you talk a little bit about those and the conclusions that you may have come to?
From the animal trial?
Yes.
So,
No, well, I was even thinking the whole series of trials that you've done so far.
oh, the whole series of trials? Yeah, of course. So during the trials, seeing the results, we could see that omitting any of the amino acids, when we remove any of them, we do have an effect on the milk fat yield. It does change something. And we could also see that some of the amino acids that I found that had the most effect on the milk fat yield was the same that Izabelle found on her previous research with supraphysiological levels. For me, I found lysine, methionine, isoleucine, and leucine being the ones that affect the most the milk fat yield.
Okay.
Yeah, I think that's what we got as big results right now. So those are the amino acids that we're using in the second experiment to analyze the different levels and the different interactions between them.
Very well. Very interesting.
Yeah, and also the ones that we're going to use on the animal trial, right?
Yeah, they're the same.
Yeah. And the idea is to see it, because at this point we already know that just addressing one amino acid time is not the best way because they interact, right? So she's designing the experiment in a way that we can see the interactions among the amino acids and see the amino acids as a group, not only as individual amino acids.
Mm-hmm. So going forward, what do you see as maybe some practical implications, and then maybe talk about some additional research that's going to be needed?
Yeah, sure. I think some practical implications. I think this knowledge is important that maybe in the future we can manipulate a diet in terms of amino acids to try to increase the milk fat yield. That would be really interesting for the producers.
Mm-hmm.
And I think the future research, we still need to explore more how the amino acids are interacting and which amino acids we should work together, which should be the groups so we have better results.
Very well. Speaking of the future, what's your future hold? You're now a visiting student here at Virginia Tech. What's next for Yumi?
You're putting me in a hard place here. Okay, so I think my future, I really want to go to grad school. I think about teaching in the future. So, yeah, I'm I have an opportunity now, and I'm still open for opportunities. Yeah, that's it, I think.
Okay, very well. So anybody out there listening, Yumi's open to other opportunities. Very well.
And I'm in the line. She already has an invitation
Now,
for
here's
grad
where I'm in
school.
trouble.
Yeah, that's my opportunity. That's my main goal. I'll be there.
I'm convincing her to go back to Brazil, finish the vet school, and then come back to do the PhD at University of Idaho.
And I'm willing to, yeah.
There you go. All right. Well, listen, I've really enjoyed the discussion this afternoon. Izabelle, thank you for joining us.
Thank you.
Yumi, it's been great, and all the best to you. Good luck. Well, whatever
Thank
you
you so
decide.
much, yeah.
All right. Welcome back to the American Dairy Science Association meetings. We're here this afternoon with Dr. Tom Overton. Tom, you're standing in for Dr. Allison Kerwin, who received her PhD last winter, but recently had a child. So she's not able to join us, but you're here instead, and we hope you can do a fine job for her.
I won't do as well as Allison could, but I'll do my best.
Yep. Tom, I brought two co-hosts with me today, Dr. Pete Morrow. Pete, give us a little bit background about yourself.
Sure, Scott. My name's Pete Morrow. I joined Balchem here in the past 6 months. I've just started my career in the last few years in the industry side after spending 16 years as a private practice dairy veterinarian.
And Jeff, give us a little background on yourself.
Well, I think you got a little background earlier, but I've been with Balchem now for 12 years, technical service, traveling domestically as well as globally.
All right, very well. Tom, the paper that we're going to be reviewing is titled "Relationship of Blood-Based Indices of Liver Health During the Transition Period with Performance and Health." What can you tell us about the research project?
Yeah, so we got into this a few years ago. So, some researchers in Italy, Bertoni and Trevisi, a number of years ago, created what they called a liver functionality index, and so that was based on things you can measure in the blood like bilirubin, cholesterol, and albumin, which represent different aspects of either acute phase response or liver metabolism. And in smaller data sets, they related it pretty nicely with, in other words, cows with poor liver functionality index, had poor outcomes anyway in transition. The one limitation that from my perspective that they had was, in what they developed, they required, sampling the same cows at like 2 and 28 days in milk. And so from the standpoint of our approach, where we want to try to, determine and develop, markers that the industry can use, there's kind of some limits of practicality in terms of doing, 2 time points in the same cow, at that time frame. So we really wanted to see if we could adapt it to a 1 time point type index.
Very well. I'll toss it over to Jeff and Pete. Dig into some of the data.
I was just wondering if you think that this could be a good predictive measure possibly in the future to know which cows maybe we should be looking after, or we think that maybe we know they're going to be successful based on these indices.
Yeah, so I think that's a great question. I mean, one of the things, at least at this point in time, amongst multiple data sets, at least relate to cow level outcomes, we do see with our single time point type approach, and we've looked at a few different ways now, that is even just a 1 time point approach is associated with, poor outcomes, whether it's health, production, et cetera, during transition. I had a grad student here with a poster, Tate Nelson, a PhD student of mine. He actually looked at different time points and then compared to kind of the liver functionality index time point. And actually 2 days in milk was actually not a bad time to make that measurement. So I think, that may offer the opportunity to at least identify early cows that may be at more risk for poor outcomes. I think we're a ways away, though, from being in a place where we're going to use it to say, okay, we're going to identify individual cows that need treatment. I think for the moment, anyway, I feel a little more comfortable with it as a herd level type assessment, where we might use it within a herd, kind of as a barometer for how things are going. Again, it's still kind of a relative measure, and so I think we can add to the data set. But I think there's opportunity there, and I think it gives a little different look than what we see with some of our traditional markers like NEFAs and ketones.
So Tom, why do you think right now there's a limit to it being a cow marker versus?
Well, I just think
What's
so.
the
So again, right? These epi studies are all about more risk versus less risk, right? So I mean, when you go over a certain threshold or things like that, I think again, as a relative marker, there's unlike with things like NEFs and ketones, right, where we can say, okay, that cow with 1.2 millimolar blood ketone, but BHBA or more, right? She's at more risk for things. We don't have a single value that we can use at the herd level to be able to say, okay, she's at, in general, it's more of a relative type basis, right? So it's a little less handy from the standpoint of being able to interpret for individual cow use at this time, I think.
So let's pretend we can find that one biomarker, haptoglobin, whatever it may be.
Yep.
And we have a number for it. What's it going to take to be able to do that on a daily basis, like we do like BHBA?
Yeah, I think one of the things too. I mean, this you look at analytes for these three markers anyway. At least as Bertoni and Trevisi, but then us, incorporated into this index. You're looking at $16 to $20 a sample, right, for analysis. So we're not going to be running this like you do BHBAs at the herd level, right?
Yeah.
I think that's pretty clear. But again, as a periodic evaluator of how things are going, right, from a health standpoint, again, give you a little different look than the energy related markers. I think there is some opportunity here going forward. We're also, collaborators also that are looking at developing some multiplex assays involving not only acute phase proteins but cytokines as well. So there's different directions here we can go. But so far this liver health index seems to hold up reasonably well, compares reasonably well with what Bertoni and Trevisi did, and we've again looked at it now in several different larger data sets, and it holds.
If you had the opportunity, or which additional acute phase protein would you add to a indice like this to maybe explore other options, maybe more, whether it be sensitivity or specificity for this type of test?
Yeah, I think again, our previous work that we've already talked about with haptoglobin, right? I mean, that one's pretty well established as there's probably no perfect marker, right? But that's probably reasonably well established anyway. Its relationships with downstream outcomes. Well, what we haven't done, and it'd be interesting to do that actually, is try to start to look at some of these things within the same data set to see if we enhance, as you indicate, right, enhance the sensitivity, specificity, predictive value, all those things that we want accuracy on, when it comes to diagnostic tests.
Would you think this system could be used as a scorecard for your transition management program?
I think potentially, right? I mean, so one of the things that to some extent you're kind of comparing it to population. So you're comparing either within a herd against itself, right, or potentially over time. If we have larger numbers of data out there, larger samples, larger number of herds collected, you can kind of look at where your herd stands relative to maybe others, right? And so I do think there's opportunity there to look at some of these things. There's going to be careful to benchmark too heavily, right? But at the same time, I think there's value in having some reference numbers and then comparing to them.
Yeah. Interesting information, gentlemen. Tom, any final words that you'd like to leave with the audience?
Yeah, no, again, it's fun here at meetings, our first in-person meetings in 3 years, and so it's fun for our students to get back out here. I've got 3 students here that have never attended an ADSA meeting before, and they all have posters, and so it's a great opportunity for them to get some exposure, and thanks to y'all for doing this.
Yeah, well, thank you for helping us out. Thanks. See you next time.
Tonight's pubcast stories are brought to you by the Keysure line of chelated minerals. Keysure and Keysure Plus deliver proven and consistent bioavailability to maximize performance, and a no-frills pricing approach for greater profitability. Visit Balchem.com to learn more.
Welcome back to the American Dairy Science Association meetings. We're here once again with Tanya France from Cornell University and Joe McFadden, her advisor at Cornell University. Tanya, you have a presentation tomorrow called, the title is Effects of Feeding Rumen-Protected Methionine and Calcium Salts Enriched with Omega-3 Fatty Acids on Lactation in Periparturient Dairy Cows. Joe, I understand that this was your idea. This was the genesis. Can you talk us to how you came up with the idea?
Yeah, no problem. there's plenty of evidence to show that rumen-protected methionine feeding in transition cows has a lot of beneficial effects on milk production and metabolic health. You know, it acts as an antioxidant that enhances something called the transsulfuration pathway that is really important to help cows mitigate the negative effects of oxidative stress, and has this ability to reduce inflammation. So that's really important as a nutritional therapy for transition dairy cows. Omega-3 fatty acids, especially very long-chain omega-3 fatty acids like those that come from fish oil, a little less work focused on their effects in transition cows. But again, they're anti-inflammatory, generally recognized as being health-promoting components of the diet. And so we want to look at the synergy between those. There's some hypotheses related to liver function and how they can work together, both methionine and omega-3 fatty acids, to really optimize liver function. And so that's one of the things we wanted to explore with this study.
All right, very well. My co-host for this session is Dr. Ken Sanderson. Ken works for Balchem. Ken, tell us a little bit about yourself.
So, hi, Scott. I'm with the company now, actually, for the second time. I've been in the industry for 40 years, but the last 3 back with Balchem in the role of global business development. And I work on a number of different projects, including some of our research work as well.
Oh, very well. Have you met Tanya yet?
I have met Tanya.
Right.
We had a chance to listen to Tanya's presentations yesterday.
All right. Yeah, excellent. Well, Tanya, why don't you tell us a little bit about the research?
Yeah, so this study was looking at basically the co-supplementation of omega-3 fatty acids and rumen-protected methionine, and we started feeding 79 Holstein dairy cows at about 3 weeks prepartum, and we followed them through to about 4 weeks postpartum, co-supplementing them. And during this time, we did weekly blood sampling, milk sampling, liver biopsies 3 times for each cow, and really wanted to measure the effects of the combination supplementation of the rumen-protected methionine and omega-3 fatty acids. The other 3 treatments were without both of those, or with just adequate methionine, or with just omega-3 fatty acids.
You know, one thing I'll add to that is that, you know, typically in the field, you're not feeding a lot of added fats to close-up diets. But we did do that in this particular study. Total fatty acids in the diet were 1.5% of ration dry matter from the close-up period through early lactation. You know, there's a lot of science that we're thinking about in terms of why we did that. And you know, one of the things that were sort of a secondary objective of the study is to investigate the effects of these nutrients, methionine and omega-3 fatty acids, on sort of the microbial composition. And doing that with some fecal samples is what we're going to do, and how that might relate to changes in gut permeability. So that's sort of a long-term objective that's not presented here at today's conference, but it's something that's on our mind, and that's sort of why we went with a longer sort of feeding duration for the supplemental fats. You know, the production outcomes were really strong. I'll let Tanya talk about that. But you know, it didn't hurt that we added it to the close-up diet.
So one of the questions that actually was shared with me about this abstract was related to the background of the other amino acids in the diet. So can you tell us anything about the lysine content, for example, in the diets, or do you have some?
Oh, you want to point to me?
Yeah.
You said you'd answer this one.
Okay. All right, yeah, so lysine was kept fixed as right around 7.1% of NP. So we did put some rumen-protected lysine products in the diets to make sure that was sort of constant across all the treatments. When you look at the lysine to methionine ratio on the methionine deficient diets, that was closer to about 3 to 1. And on the diets that had the supplemental methionine in it, that ratio went to about, I believe it was about 2.5 to 1.
Excellent. So there's been a fair amount of previous work looking at methionine and showing increases in dry matter intake in the postpartum period. And I guess we're curious about the mechanism and what you propose is the mechanism around that change in intake.
really we think it is more driven by the increase in milk production, which would influence their dry matter intake postpartum. So feeding adequate methionine is typically going to help partition more nutrients towards milk protein synthesis or just yield in general. So that being said, those increases in dry matter intake that you're typically seeing, we're suspecting it's due to increases in milk production from feeding rumen-protected methionine.
Yeah, the only thing I'll add there is to sort of say that, and if you look at the data, and we actually whenever we get it published, or if you look at the abstract, perhaps I forget what we wrote. The diets that were lowest in dry matter intake were the diets that were inadequate in methionine and unsupplemented with calcium salts containing omega-3s, meaning that the diets that had omega-3 fatty acids in it, but no methionine, they actually had greater intakes than those that were completely deficient in both nutrients. So I think I know what you're talking about, that there is this sort of consistency that methionine feeding does increase dry matter intake, but take that with a grain of salt, knowing that that was only observed in the completely deficient diets of both nutrients. Yeah.
Okay, excellent. You got a really significant production response, and I guess I want to hear a little bit about the synergy that you saw between the methionine and the omega-3 and a proposed hypothesis around why the synergy.
So I think that, and like Dr. McFadden said earlier, with evidence of improved anti-inflammatory responses with omega-3 fatty acids, I think there's something there with adding the omega-3s, kind of mitigating this inflammatory response in early lactation, kind of driving some of these improved production performances. So just having an overall healthier cow, healthier liver function, that could kind of drive utilization of nutrients in general towards higher production responses.
Yeah, the only thing I'll say to that is that look at actual milk yield. There was no effect on milk yield. It really came down to energy-corrected milk yield. And so with the co-supplementation, that's where we saw the maximum benefit. It was over 5.5 kilos, I believe, right around there in energy-corrected milk and fat-corrected milk. I'll say that just supplementing the omega-3 fatty acids or just methionine also had significant gains in energy-corrected milk yield. So it was really the omega-3 fatty acids surprisingly driving milk fat production, and the amino acid methionine driving milk protein production. And so those two together sort of really optimized the response. Now, there's certainly a health component, and this study's ongoing in a way because we're still doing a lot of the sample analyses. She definitely mentioned this sort of inflammatory component that we want to explore, but we're also exploring liver function. One of the other abstracts she was looking at was liver functionality, sort of measuring it using an indirect approach. And co-supplementation of the 4 treatments had the best estimated liver function. And so we got to dive a little bit deeper into that to better understand the biology.
Yeah. Tanya, as we kind of wrap this up, can you kind of give us a couple key takeaways from the research, and then where do you see it going from here?
Yeah, so I guess one of the key takeaways I would say is people are kind of intimidated by fish oil supplements and dairy cattle because it's been shown to decrease dry matter intake and those kinds of things. And we were feeding at about 6 to 7 grams per day postpartum for EPA and DHA, and we saw these responses. So I think that's kind of a stepping stone and very long chain omega-3 fatty acid supplementing, considering there is no requirements or established feeding rates in dairy cattle at the moment.
Yeah, like this study we're talking about adequacy of methionine because we have some established requirements. When we think about omega-3 fatty acids, and especially very long chain omega-3 fatty acids like EPA and DHA and fish oil, it's sort of uncharted in terms of the amount that we should be feeding. I think this is at least a start in the right direction, and hopefully many more studies to come where we can tease that out. But because there are a variety of rumen-protected forms of these products on the market, calcium salts being one, but there's lipid encapsulated versions, there's versions with starch-resistant matrix. There's different types of encapsulation technologies that are out there, and so they're going to be available for farmers and nutritionists to use, and we need to know how much to feed. And what really complicates it is that I could talk about this forever, but omega-3 fatty acids also include 18-3 alpha-linoleic, and that would be the most predominant alpha-linoleic omega-3 fatty acid in the diet. But we don't know the relative sort of efficacy of an 18-3 versus the EPA and DHA. And so there's a lot of science there that we need to figure out as well.
All right, more research is needed.
Yeah.
Tanya, thanks for joining us here today. You've a very bright young lady. Your future is bright, and we thank you.
Thank you so much.
Welcome back, everybody, to the American Dairy Science Association meetings. Joining me today is Dr. Jonas D'Souza. He's with Purdue Agribusiness, all right. And we've also then got Jair Perales-Giron. He's a student at Michigan State University. And my co-host today, Carrie Estes.
Hello.
Good to see you. Good to have you back. Jair, you're going to be talking to us about, was it a paper or a poster?
No, it's a poster. We did a presentation this morning, so it was a good presentation. A lot of people came for asking questions, so I guess we are going to continue answering some questions.
Yep. So the title was Determining the Relative Metabolizable Methionine Content of Rumen-Protected Products and Their Effects on Production Responses. So give me an idea of the genesis or the inspiration for this trial.
Yeah, I guess you can see this study as an idea to try to identify the bioavailability of 4 different products for making decisions at an industrial level and as a company. So the idea was trying to use a methodology that is not that complex compared to other methodologies that has been developed before than this one, and try to do it as easy as possible for for making those decisions.
Okay. And Jonas, what was your role in this study?
So at Purdue, we use a lot of and give recommendations in terms of how we drive milk fat and milk protein yield to the dairy industry, and of course methionine is a big part of that, of those recommendations. And I can tell you every time we see it, you have these different methionine products in the market. So people ask, what's the bioavailability? Do you think this number is really what it is or not? So from our standpoint was, well, let's try to test, from an university standard with high producing animals, what is the relative bioavailability of some of the products that are in the market.
Okay.
So that's what we try to do.
And how did you decide which products you were going to test?
That's a good question. So it has, when we start thinking about, we decide to choose, basically you have different encapsulations and protection methods today, right? So you have the pH sensitive polymers, you have the encapsulation with different fatty acids. So, you're always concerned about, how you really determine the bioavailability in some of these different methods, different protection methods. So that was kind of the basis for us. So we choose 2 pH sensitive products and 2 that are basically fat coating methods on protecting that. And also, we want to use relevant brands in the market that people are using. So in a commercial situation, we have relevance to the end user, that information.
Right. Very well. And Jair, what was the basic protocol? How'd you set the trial up?
First, we have to choose some mid-lactation cows. So we did a preliminary period. So we worked with 36 Holstein cows, and we have multis and preen cows. So we selected them, and we grouped them by their milk production during the prelim period. So we had 4 cows per group because we did a Latin square design, and we have in total 9 squares. So we selected them by the mean yield, And during that period, during the prelim period, we feed them a selenium source for having a constant selenium flow to the mammary gland and assuring that the selenium was going to be the same for all the cows.
Got it.
Yeah.
Very well. Carrie, looks like you've got an
Yes, I
question.
do. Yes. So, as you know, feed and mixing stability, that's a pretty crucial piece for determining bioavailability of any rumen protected product. So, how did you handle these products before they got to the cow? Did you mix them?
Yeah, we know that that's a really hot topic about how are you gonna feed the methionine to the cows. So we did a mix. We used ground corn for putting all the supplements in ground corn, and we used that mix in the TMR. So the idea was giving the mixes and giving the products to the cow in a constant manner. That's why we did that.
And you fed once a day?
Once a day.
Okay. Okay. Yep.
Yeah, just I think your question is really relevant in terms of methodology. We did try this trial to be as close to the field as you can, right? So what we try to, but we also know that some methionine sources, you may have some fragility issues when you run through some special mixes. So basically, what we did is a 4-pound premix of that methionine source, and then it was mixed again on the TMR, and then fed once a day. So the idea is let's deliver the methionine at closest to the field conditions as we can. So that was one of the objectives.
Yeah.
And we think, depending on the methionine source that you're using, maybe you're penalizing some sources or other because of their method of delivering. If you're using post dose, or if you're using like a straight top dress type of mixture. So that's why we decided to do that way.
Yeah, it makes sense. Yeah. And then my other question I had was about the technique itself. So, as I know you all are aware, there are so many different methodologies, right? Especially for amino acids to assess bioavailability. So what about this technique makes it, I want to say, superior to the other techniques that are out in the industry today?
I guess the most important part about the technique is how easy it is to run it. Because the only thing that we need to do for developing and for running the technique at the farm level is feeding the selenium source during an estimated time. And after that, we are going to be analyzing the milk of those cows for selenium, and we are going to be doing total nitrogen. And those kinds of analyses that are really easy to do in a lab. So compared to other techniques, the other techniques involve the use of in vitro bags and the washing. And some of the other techniques involve using a radiolabeled methionine, or you need to bleed the cows. So we think that with this one, the only thing that you need to take and collect is milk. And that's something that you are doing every day at the farm. So for us, we think that that's pretty convenient.
So do you think people could run this technique on the farm, like not in a university type environment?
Yes, I think it's doable, but of course people need to do some training because we are working with selenium, and the cross-contamination with minerals is really easy when we are talking about samplings and samplers. So the only thing that I need to be pretty careful is about cleaning the samplers in the most proper way for avoiding that cross-contamination and for having the sample that we want.
Jonas, I think your company uses this methodology to make some business decisions. So I'm going to guess you have some level of confidence in it. Would you mind give us an idea of how consistent do you think it is? How precise is it? We'll speak to that a little.
Very, very good question. Carrie really is trying to get us in trouble here when she asks us to compare techniques, right? But I think the point is understanding the technique has its value when we try to disrupt less that normal feeding schedule, right? It's a less invasive procedure. Like when you start bleeding cows too often, how you change those partition of amino acids and how how in terms of immune system, how really it affects the animal. We don't know, right? We made all these assumptions. So I think that's the benefit of the technique. In terms of making business decision, one thing that we always wonder and we try to do in this trial is not only what that bioavailability is, but what's the variation around that bioavailability. Because you can have a bioavailability of 50%, it varies from 10 to 90, you might be in trouble, right? So that's, I think, is another good point of what we try to do here. We try to use more cows, using 36 cows, and Latin square gives you a lot of power. If you see what we did is try to estimate that error using Monte Carlo simulation. So in terms of business decision, I would say don't look only to that bioavailability number, but look to the variation around the bioavailability number when you make decisions. And I think that's pretty important, and that's what we try to assess with this trial here. And that's from basically, if you think when we set up the trial, we use as payload the manufacturer recommendations, and you can see the response of the animals in the bioavailability estimate. So I would say from the sources that you use, 3 are pretty within what we expect. The manufacturer recommendation, only one source would be out of the recommendations based on these experiments, right? So the technique allows that, and I think not only evaluating the bioavailability, the relative bioavailability, but what's that range is really the key.
Yeah. So just a real quick question. I believe one of the treatments was AminoSure XM, of course, marketed by Balchem, and so I don't want to talk about the other ones, but can you tell us, was AminoSure XM one of the 3 that did meet the manufacturer's requirements?
It was. Yes, and you can see the error as well. This was within recommendation. So I think the technique allows us to do that quite well. And as I said, in the feeding situations that we did, I think when you use a fat protection mechanism, maybe sometimes you are penalizing that by using different methods. So, and basically, it was one of that was within your manufacturer specifications.
Good, very well. I think Clay did a good job when he put those specs together.
Oh, he's on track.
He's on track. All right, very well. So, any other key learnings, key things you'd like to share with the audience? Wrap things up.
I guess this is a technique that is really easy to use. If you are gonna want to see, as Jonah said before, the consistency of the supplements that you are going to be using at your farm, this is really easy to do because, as I told you before, selenium or nitrogen are kind of a common analysis that some labs used to do. And as I told you before as well, milk, cows are producing milk every single day, and that's the sample that we use for running this method. So this is something that is really easy to do, and of course it's gonna have all the outputs that Jonas told us. That it's gonna give us a clearer scenario about the quality and the consistency of the products that we are going to be feeding to our cows.
Yeah, excellent. Well, listen, I want to thank both of you guys for stopping by, spending some time with us today here at the Real Science Exchange, and I hope to see you next time here where it's always happy hour and you're always among friends. We're not drinking today.
Thanks for having us, and it was my first podcast, so yeah.
Yeah, good.
Happy to do it.
Well, let's hope it's not your last.
Yeah. Okay. Yeah.
All right. Thank you.
Okay.
Thank you.
Hello, everyone, and welcome back to the American Dairy Science Association meetings here in Kansas City, Missouri. With us today is Kayla Allward. Kayla is a PhD student at Virginia Tech by way of Georgia. Mm-hmm. Welcome. Glad to have you here.
Yeah, thank you for having me.
And you're going to be, are you presenting a paper or poster?
Presented an oral on this. Yep.
Okay, presented an oral, and it's the dry period environmental impact on colostrum volume and quality. Sounds real interesting.
It is. I'm really excited. Glad you guys asked me here.
Yeah, super. So why don't you just give us a little bit of background on the genesis of this project?
Sure. So I think this idea for this project really came from some anecdotal information from producers who were, kind of unsure of why they're seeing some slump in colostrum volume in fall months. trying to figure out why are we seeing this slump. And so then we started thinking into, okay, what could be impacting the cows that we're not already accounting for? That the dry period's managed pretty closely. I mean, we're really strict on how long they're dry, the vaccination protocol and everything to make sure we get good colostrum. So what factor are we not taking into account? And we thought that was going to be seasonality. And so that's kind of led us to this project of trying to figure out, okay, during that dry period when those cows are dry, is the weather what the missing factor is? And so that's kind of the basis for this project.
Yeah, very interesting. Kayla, I've invited a couple experts with us tonight. Dr. Pete Morrow, he's a DVM. He works for Balchem Corporation. Also, Dr. Clay Zimmerman. He's my co-host on the Real Science Exchange. Clay, I saw you shaking your head over there, agreeing with producers seeing a slump in the fall. What do you think about that? Is that something you've seen as a practicing nutritionist?
Yeah, yes, we get questions about that all the time. Yes.
Good deal. So, Kayla, why don't you just give us an overview of the project, protocol, and how you put it together?
Yeah, sure. I'd be glad to. So this project was actually conducted at 3 different farms in Virginia. We tried to get a lot of animals enrolled in this project so we could get a really robust data set. And so we ended up enrolling 3 different farms. We had I think max about 150 animals from one farm, and at the minimum about 50 animals from each of the other farms. And so what we did was when these animals calved, we had the farm measure with a Brix refractometer, kind of an on-farm estimate of colostrum quality. They also measured colostrum volume as it was produced, and then they took a 50 ml sample for us, and we sent that off to DHIA to be analyzed for components. And we also looked at IgA, IgG, IgM, those important immunoglobulin levels. And then in addition to that, where that photo period and kind of THI thing comes into play is after we collected all that data, I went back and found what the nearest weather station was to each of those farms. And then for each day that the cow was dry, figured out what the average length of daylight that she was exposed to was, and then also figured out what the average daily THI, or temperature humidity index value, was. And then figured that out for her whole dry period, came up with an average for her far off period and the close up period to try to make some associations with, what was going on with weather and how that impacted their colostrum volume and quality.
So were the dairies, were they very far apart, or were they?
Yeah, so we tried to capture a range, and we got some dairies in Northern Virginia, some right near us in Southwest Virginia. We got a wide range.
Interesting.
Yeah.
From your studies, you think you could take and be a practical application in terms of, talking about day length? You think we could do some day length manipulation to improve colostrum production and quality?
Yeah, that's a good question. It doesn't seem that the photo period seemed to impact colostrum quality and volume much. It seemed like the temperature humidity index was really that more important factor. And so it's already in place, the recommendation for dairy farmers to limit the amount of light that cows are getting while they're dry, because that's going to improve milk production after they calve. And so we kind of wanted to see could that be having negative impacts on colostrum? And we didn't see that. So it seems that THI is the more important factor. And when we started getting cows that were up in those higher THIs, they were having negative impacts on their colostrum volume and quality. But of course, that doesn't really jive with what producers are seeing. They're saying they're seeing a slump in colostrum in the fall. So I've still got some more research to do with that to try to figure out why they're seeing that slump and we're not in our data. But it does seem that THI is the more important factor. And what's more interesting is that it was only for the close-up period. So all the data that we collected on the far-off dry period, so anything from the day they went dry until 21 days before they calved, didn't really have any effects on colostrum. It was just that close-up 21-day pre-calving period that was real critical.
So, Kayla, were the samples were taken all throughout the year?
Yeah, so we collected data for about a year and a half. So I think it was something like November from one year, all through that next November, and then into July or August of that next year. So not quite 2 years of data, but yeah, quite at least one full year to capturing that whole season.
Have you had the opportunity to look at anything besides immunoglobulins, maybe into the cellular content of the colostrum? Is that something you've evaluated?
Good question. So we did not evaluate that. I do think it's interesting, though. We've got some calf data. So we do have some birth weights and some calf health up to 60 days, so that pre-weaning period. And I haven't had a chance to analyze that data yet, and it's on only a subset of those animals, but I'm really excited to see what that data looks like, to see if we can make some associations with, seasonality and that calf health factor.
So colostrum yields were measured in these
Yes,
cows?
yeah. So the farmers did measure the colostrum volume from that first milking.
So how soon after calving were they harvesting colostrum?
Good question. So it ranged a little bit farm to farm, but they had pretty strict procedures in place, and they did have a night shift crew on all of those farms. So it was within 6 hours of calving. So we tried to get pretty right after calving to get that first measurement.
Do you have recorded data maybe to how the time from calving until the colostrum is harvested, or was that not collected?
We do have that, and we also have the time of day that the animal's calved and that sort of thing. We did have a couple issues with maybe some differences farm to farm with not writing down the exact time, so we don't maybe trust that data quite so much. We did make sure that everybody was collecting everything within that first 6 hours, but some of that other data that the farmers were putting in might be a little bit off, so I haven't included that in any models yet.
Were these, did these dairies, do they have adequate cow cooling for the dry cows?
Yeah, great question. So what were those cows doing during their dry period? All of them were housed on pasture. So that's an important thing to note. We did check and make sure before we kind of collected this data that all of them had similar dry cow procedures. So, similar dry off procedure, dry off date before their due date, and similar ration that they were fed. And as far as cow cooling, they were similar. Some of the farms that were in the southern region did experience some higher THI values. So our results do vary pretty significantly by farm, and we had to keep that in the model. So in the future, I'd really love to redo this project and do it on one farm where I can get a whole bunch of animals. But just for, to get animal numbers, we ended up collecting data from 3 different farms. But yeah, things vary pretty significantly by farm.
So you talked about the future, wanting to do it again. Are you going to be able to? Do you have enough time?
Well, that's a really cool question. So we actually did our own study on cows at Virginia Tech, where we housed them completely in a temperature-controlled environment and just manipulated a photoperiod to try to tease out, is it photoperiod, is it THI? And that's another abstract that I included, but I can share with you guys that there wasn't any differences by photoperiod. So when cows were exposed to either a long day of 16 hours of light or short day of 8 hours of light per day, there was no effect on colostrum volume and colostrum quality that we've seen so far. And we did that study with Jerseys and Holsteins. So it's pretty interesting that we're not seeing that difference there. That's what leads me to believe maybe temperature humidity index is more that important factor.
So in your photoperiod study, did you see effects on milk production beyond colostrum?
Yeah, great questions. We did. So we followed those cows until they were, I think, 15 weeks in lactation, and we did see that the animals that were limited in photoperiod produced more milk after they calved. But there's a little bit of a conflicting data. We didn't see as much of an increase in Holsteins. We saw a big difference in our Jerseys, not as much in the Holsteins. So we're still trying to tease out, what are some breed differences, because these guys are definitely behaving differently after calving.
Kayla, as we wind down here, do you have any final thoughts you'd like to share with the audience?
I think this is a really cool project. I think it highlights that we're really closely managing colostrum as an industry and looking at a lot of cool key factors. And I think most of them have been explored and kind of dialed into what we need to make those the recommendations for them in order to perfect colostrum volume and production. but I still think that there needs to be some more research in photoperiod and temperature humidity index, and particularly how they differentially impact different breeds of animals. I think there's some information out there that we haven't yet collected, and so I'm excited to keep doing some research on that and get some more information so we can make more recommendations and improve colostrum for the industry.
Well, thank you for stopping by today, Kayla. You're a very bright, very impressive young lady, very articulate. What's the future hold for you? You're in your third year, PhD student at Virginia Tech. Where do you go from here?
Yeah, so I'm excited to be writing a postdoc grant. I'm hoping I'm going to stay right at Virginia Tech. I'm going to switch gears a little bit and get into reproductive physiology. So that's what I did for my master's, and that's what I'm hoping to get back into for my postdoc. And then I'd love to go into academia, teach students, have a role in extension and helping producers, and continue conducting research.
Oh, Clay, I was hoping she'd say she'd wanted to go to industry, because I think we got a good one here. So.
That's right. Yes.
Well, Kayla, I appreciate you stopping by. Appreciate all the research that you've done, and wish you all the best.
Yeah, thank you so much. Thank you for having me.
All right, very welcome. Hello, everyone. We're coming to you from the ADSA meetings here in Kansas City. With me is Miguel Cherivi, right? Yeah,
correct.
And Andreas Contreras. Almost rolled my R. Anyway, they are from Michigan State University. Miguel is a third-year PhD student, and Andreas is a advisor and associate professor there at the university. Miguel did a presentation, I believe it was yesterday, an oral presentation, and the title of his talk was Lipolysis Inhibition Improves Clinical Outcomes in the Treatment of Ketosis in Dairy Cows. So very interesting. Had a conversation just earlier and found that very interesting. Why don't you give us an overview of the work that you did and the presentation you gave yesterday?
Okay. First of all, thank you for inviting us. It's great to be here with you. The main idea of this clinical trial was to enroll clinical cows with clinical cases of clinical symptoms of ketosis, and those cows were from a commercial dairy. So the idea with this study was to not just treat the ketosis in the terms of improving glucose concentrations, but also we want to inhibit the increased lipolysis that is occurring in those cows.
Okay, very well. And then, Andreas, what was the protocol for this trial? How did you set that up?
So the idea was to use the standard treatment that the commercial house use for treating clinical ketogenic cows, and that's propylene glycol. And as Miguel said, that propylene glycol really targets the hypoglycemia in those cows.
Yeah.
But also we wanted to include lipolysis inhibitors, because as you know, lipolysis dysregulation is one of the key leading factors to the development of clinical ketosis. So we use two inhibitors. One is niacin, that targets the classic lipolytic pathway, and the other one is banamine, which targets the inflammatory pathway for obvious reasons, right? It's an NSAID. So we enrolled about 110 cows, Miguel, and we divided them in 3 treatment groups, and those treatments included the glycol, the standard treatment, glycol plus niacin. It is the rumen protected niacin from Balchem, and the third treatment was propylene glycol plus niacin plus the flunixin, meglumine or banamine. And we also enroll control cows that were matched by days in milk and lactation number. And we started the treatments, and can you please describe the treatments, Miguel?
The treatments?
Yeah, the dosing. And
okay, correct. So propylene glycol, it was the dose that had been used for the dairy farm. It was 300 grams of propylene glycol once per day for 5 days. Niacin was administered 24 grams once per day for 3 days. And the cows that received the flunixin was administered 1.1 milligrams per kilogram once per day for 3 days.
And just kind of for clarification, I think that was 24 grams of the rheumatoid-protected niacin,
Correct.
not just straight niacin.
Correct.
Yeah, very well. I'd like to introduce my co-host for the session, Dr. Glenn Ains. He's Technical Service Manager for Balchem. Welcome,
Thank you.
Glenn.
Appreciate it, Scott. Always good to be here, buddy.
Yep, it is.
Just a quick question. So, I've never heard or I guess knew much about Banamine in terms of impacts on lipolysis. So, I understand how Niacin works. Method-wise, how does Banamine work?
Okay, so we recently demonstrated in a paper that just went out recently that inflammation is important in controlling lipolysis. So in this paper we start looking in the effect of LPS as a model of inflammation. So we consider that inflammation is an important factor regulating lipolysis. So when we move to the Banamine, so there are for sure as an anti-inflammatory drug, it could target not just the endotoxin that may be present, but also cytokines or TNF-alpha that are triggering lipolysis. But essentially, the main factor that is reported in other species is the reduction of the prostaglandins by Banamine is one of the main targets of reducing this lipolysis, because it's demonstrated that prostaglandins are also targeting the lipolysis within the adipose tissue.
Interesting. Okay.
So I think is that when IL-6, for example, which is a cytokine, and TNF-alpha are secreted by immune cells or by the adipocytes in the fat tissue, those two actually trigger lipolysis, and it's an independent pathway to the classic pathway, which is the one that's triggered by adrenaline or growth hormone.
Mm-hmm.
So when the interleukins bind to a receptor in the adipocyte, that triggers actually the activation of lipases. So the idea was to target the two triggers of lipolysis in the adipocyte in cows that were clinically ketotic. So that was the goal here.
In anticipation that they would be sort of additive.
Yeah.
Yeah, which is why you did the propylene, then the niacin, and then the propylene and the niacin and the Banamine.
Correct.
Okay. Yeah, that makes sense.
Mm-hmm.
And so in terms of how you evaluated the cattle and their response to it, can you address that?
Correct. So we, as the Contreras mentioned earlier, so we have some criteria for inclusion. So we established that a cow that were recovered also when they were free of clinical symptoms, when they recovered the feed intake, and most important when BHB levels were lower than 1.2 millimoles. That was the point for inclusion. So we collect specifically samples on day 3, day 7, and day 14, and in those days we evaluate the rate of recovery of those cows.
Okay. And what did it tell you?
So essentially we start observing immediately at day 3 a better recovery in the cows that receive the niacin and also in the cows that receive niacin plus flunixin, being better for the cows that receive the combination of niacin and flunixin. And specifically on day 7, it was statistically significant, this difference, and we observed that the combination of this niacin and flunixin had 1.4 more chances of recovery than a cow that received just the propylene glycol.
So you saw the additive effect that you were
Yes,
looking
yeah.
for.
So if you wanna look at it in practical terms, the lowest recovery rate was for glycol alone. Intermediate was for glycol plus niacin, and the faster recovery rate was for the cows that had Banamine and niacin in the treatment. We evaluated also the reduction of lipolysis by measuring NIFAS and BHB.
Mm-hmm.
And we also, when we collected adipose tissue explants, we evaluated how responsive was the adipose tissue to lipolysis and how responsive was to insulin. Because one of the reasons why lipolysis becomes dysregulated is because the adipocytes do not respond to insulin, which is an inhibitor of lipolysis. This is the major inhibitor of lipolysis.
They become insensitive, basically.
Yeah. So when we evaluated that, the adipose tissue explants from cows that were treated with niacin and banamine responded better to insulin than the explants that were from cows that were only treated with glycol.
Yeah, so the biology follows the logic, right?
In this study.
I know that doesn't always work that way.
I hope in the follow-up studies it does too.
So if you were to just think in terms of, I think we talked earlier that when you looked at the cattle, they had clinical signs, they were down, they were lethargic, all those fun things that you get to deal with when you've got a clinically ketotic cow, and then you treated them. And then from a physical perspective, just observationally, did they recover very quickly? Did it take a few days?
Very quickly in the cows that had both inhibitors. The herdsmen at this dairy, they were very happy with these treatments because they say, well, you got the magic bullet. The next day they are fine and they're eating a lot and they return to production faster than the cows that were only treated with glycol. So they were pretty happy at the farm.
I'll bet, yeah. One more headache eliminated,
Yeah.
or at least reduced.
Well, at least for the clinical ketosis. Right?
Yeah. So what's next?
So we want to actually explore this more in detail to see what happens when you include NSAIDs in the lipolysis control within adipocytes. Because this really has something to do with the way maybe prostaglandins, maybe interleukin receptors actually modulate lipolysis. So what if we reduce the production of those interleukins, of those cytokines? What's the impact on adipocyte lipolysis? So that's one thing. And we also want to include, obviously, niacin in the equation because there is an additive effect that's clear. So those are the next steps with this.
Yeah. Well, you're probably somewhat familiar with Reassure, or protected choline product, which can help move fat out of the liver, right? So maybe that would help as well. I'm just tossing out some research ideas for you.
Obviously, this research this experiment was focused exclusively on the adipose tissue.
Yep.
We need to include the liver in the equation, obviously, and then some treatments that certainly would target the liver, the hepatocyte.
Sure. Yeah, lots of interest in the research world over this whole inflammation thing. It's huge.
Oh, I agree. Yeah.
Gentlemen, this has been a very interesting conversation. I'd like to go on a bit longer, but we have our next interview up next. So would like to say in closing, Miguel, third-year PhD student, what's the future for you?
So essentially, first to finish my PhD.
Good idea.
It's a good idea. Yeah. And then I would like to keep doing research. So for sure, I would like to do a postdoc, and then let's see what happens.
All right, very well. Well, the best of luck to you. Andreas, thanks for stopping by. Appreciate you guys spending some time with us this afternoon.
Thank you very much for the invitation.
All right.
It's been a pleasure. Enjoyed it. Thank you.
Hello, I'm Scott Sorrell here at the ADSA meetings, and we've been visiting with students and postdocs and researchers and the like, talking about the research that they've done recently. With me is Susanna Räisänen. She's currently at the University of Helsinki. She's doing a postdoc there, but she did her research at Penn State University. Susanna, welcome to the Real Science Exchange.
Thank you.
Hey, would you mind giving us just kind of a brief overview of the research that you did while there at Penn State?
Yeah, so my main focus was on histidine requirements for lactating dairy cows, and I also did some research with bioavailability of rumen-protected amino acids, but mainly on histidine.
Okay, very well. Yeah. And with me, my co-host, Dr. Clay Zimmerman and Dr. Glenn Ains. Welcome.
Good to be here, Scott, always.
We'll let you guys dig into the data.
All right. Glenn, you want to start us out?
Well, can you tell us a little bit about your research?
Yeah, so this particular research, we kind of combined all the histidine work that has been done so far. A lot of that work was done at Penn State University, including my dissertation and PhD work. And in total, we were looking at around 20 experiments overall published between 1999 and 2021, more recent research. Some of the experiments were done with grass silage diets and some with corn silage diets. And then and also the way the histidine was supplied was both with rumen-protected histidine, infused histidine in different ways. And then we were testing, kind of simply just looking at the effect of histidine on supplementation on different production variables. And then we also did some regression analysis looking at how the responses were related to digestible histidine supply overall.
So how many of the studies were done with rumen-protected histidine?
12 of the 20, so a little over half of them. Yeah.
Did you see any difference in response between how the histidine was supplied?
We did for the milk true protein yield. There was a difference looking at the histidine type. So with infused histidine, the response was greater, or the magnitude of change with increasing digestible histidine supply compared with the rumen-protected histidine. Both increased milk protein yield as the dose increased, but more so for the infused histidine. And similarly also with the plasma histidine, we saw a difference there. And I think that's partly due to the differences in bioavailability estimations for the rumen-protected histidine. So it's a little bit trickier to have the exact dose, so probably we underestimate the dose from the rumen-protected histidine.
So what were the mean responses that you were seeing from a production standpoint with histidine supplementation?
So for milk yield, energy-corrected milk yield, it's I can't remember the exact numbers, but it's around 0.5 kilogram per day, what we saw. And then for milk true protein yield, also the response was around 0.5 kilogram per day when supplemented compared to control. Yeah. So it's pretty solid data, I think, showing this effect of histidine and importance, especially for milk protein yield.
Does stage of lactation of these animals, does that play a role?
So most of the experiments, I think, have been done with mid-lactation cows. So I don't know of any research done in transition cows, early lactation cows. So I think there is more work to be done there for sure. Especially with, we know that there is a mobilization of histidine during early lactation, and histidine is an important part of the muscle tissue also. So there might be something there to look into.
I noticed you called out the forages that you had in that research. Was that something that you were looking for, or did you try to tease that data out?
We haven't gotten there yet with the analysis, but we want to look into it. Actually, histidine was found to be the first limiting amino acid like 20 years ago at this current university in Finland. They did some work with infused histidine, and they were like the first showing that histidine could be limiting. And this was done on grass silage-based diets. There aren't any work done with rumen-protected histidine with grass silage, which I hope to do in the future, continue this work. And they haven't done any work since with histidine in Europe or with grass silage-based diets. So I really want to explore that more.
What type of grasses?
It's mainly Timothy grass, which highly digestible, a lot of soluble fiber in the rumen. So I think that's in grass silage diets. I think it's specifically due to the dependence on microbial protein, and that's why histidine probably becomes limiting in those diets.
So in all these studies you evaluated, did you look at potentially lysine and methionine as potential modulators of a response to histidine?
Yeah, we have the data. That's also something we are still kind of running the different analysis and figuring out what to include in the model and what relationships to look into. But that's definitely one important thing, is to look at I think we will go for like efficiency of utilization of different amino acids, essential amino acids, including methionine and lysine, and then looking into how the response varied depending on their efficiency in terms of histidine efficiency and supply Yeah, and it's important not to forget these other amino acids because you know it's
Absolutely.
not just one thing that's limiting, but we have to look at the profile of amino acids.
So I had a question about you showed the responses to infused versus the responses to the RP fed.
Yeah.
Is there anything can you calculate out of that or estimate out of there what the bioavailability actually would be for the
Yeah.
RP lysin?
Yeah, and I think that's something someone else also commented on this after my presentation, and I think it's a really important point and good point to. This is a good chance for me to look into the bioavailability estimations. Some of the amino acids, rumen-protected amino acids, are not the same between the experiments, so there might be. I have to look into and go back and see the estimations used in different experiments because it's like in some of my experiments I used my own estimations of bioavailability, and then others have used different, and that's also the whole another problem or thing that we have to look into is the bioavailability, especially when figuring out requirements of amino acids, and then if we are using rumen-protected amino acids, but then we still are debating the bioavailability of these amino acids and how to measure it, and then
Yeah, absolutely.
So it's a little bit tricky, but I think there is something there that I could look into and see if I can, correct for that bioavailability estimations with the plasma data.
Because you did try to estimate the digestible histidine.
Yeah, I did.
Yeah.
This was one of a separate trial that I did.
Okay.
Yeah.
So that explains the answer to my question.
Yes.
So, Susanna, with all the research that's been done now with the meta-analysis, is there enough information to estimate a requirement for histidine?
This is always a tricky question if I want to set a grams per day requirement or not. But we have been looking into the efficiency of utilization, and with Dr. Lapierre, she's been estimating that, and then I also did my own estimations in my all my experiments. And it looks like it would be around 0.77, 0.8. And based on the meta-analysis I did, it looks like it's somewhere around 60 grams per day of digestible histidine. That's the data we have, and that and the meta-analysis is showing. But of course, it depends on so many things. So yeah, I don't want to give this type of recommendation. But I think we do need some more data to establish it, and with different diets, especially grass silage based diets, low protein diets, maybe it's different. Yeah.
So in Europe, if a diet's deficient in histidine, how would you supplement it right now?
It's very tricky because they can't use blood meal, for example, which is a good source of histidine. So we have done some work with faba bean. And rapeseed meal actually has a pretty good histidine concentration. So they are saying also that if we have rapeseed meal or canola meal in the diets, they maybe are not that deficient in histidine.
Okay.
So it depends on the diet and how high in protein you want to be.
Right.
Yeah. But I want to be low protein diets, and then you have to find a way to supplement histidine for sure.
Suzanne, it's been a very interesting discussion. To kind of summarize things, what are a couple things that the audience should take from your research?
I think we have to start thinking more about histidine, included in the limiting amino acids with methionine and lysine. I think that's important, especially if we are driving toward lower protein diets overall. I think histidine is one of the three for sure to look into, and I hope we can start testing it with different type of basal diets and combining with different basal diets.
Very well. So now that you're back in Europe at Helsinki, what kind of research are you doing now?
We have been working on legume silages, red clover silage and faba bean, both as a whole crop silage and protein concentrate. And there actually methionine is one of the limiting amino acids in legumes. Okay. And that's what I've been working on, looking at supplementing methionine on legume-based diets.
Yeah, very well. Well, listen, I want to thank you for joining us today. This has been a treat.
Thank you.
All right. Good luck to you.
Very much.
Thanks.
Thank you.
We'd love to hear your comments or ideas for topics and guests. So please reach out via email to anh.marketing@balchem.com with any suggestions, and we'll work hard to add them to the schedule. Don't forget to leave a 5-star rating on your way out. You can request your Real Science Exchange t-shirt in just a few easy steps. Just like or subscribe to the Real Science Exchange and send us a screenshot along with your address and t-shirt size to anh.marketing@balchem.com. Balchem's Real Science Lecture Series of webinars continues with ruminant-focused topics on the first Tuesday of every month, monogastric-focused topics on the second Tuesday of each month, and quarterly topics for the companion animal segment. Visit balchem.com/realscience to see the latest schedule and to register for upcoming webinars.